Hello, everyone. It's a very interesting topic that we're going to discuss today on biomolecules. And the title of this presentation is an interesting one, which actually encompasses the whole idea of what biomolecules are. So they technically are the ones which try to maintain the balance of our molecular dance, the symphony of life's molecular dance. So let's start with a very important question. What are biomolecules? So if you look at the name itself indicates that probably these are molecules that are associated with life. That's why we call them as biomolecules. So these are organic molecules which are very important for life. Now, when we say life, understand this is a basic learning you all must have done. What is the difference between living or non-living things? Now, we know that living things have the capacity do a lot of activities, metabolic functions and cellular functions. If we start naming some like reproduction, growth. So this is a classic definition we all must have been studying ever since we were children, right? Living organisms show reproduction, growth, locomotion. Whereas in non-living things, you don't see any such activities. But if you go into the details of what non-living things are made up of and what living things are made up of, you will know that there are 6 basic elements. So these 6 basic elements are carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur. Now, surprisingly, most of the non-living substances you take up are made up of carbon, hydrogen, phosphorus, sulfur, but those elements that are present in non-living things are not able to create the same symphony that they are creating in living things. Why is it that so? That's probably because these chemical interactions that take place between the elements in living cells results in the production of these miraculous molecules called biomolecules, and they are the ones which are able to drive the entire living process. So these biomolecules are ultimately very important, and their structure, their chemical interactions, and their intricate designing is very important for the existence of living cells, starting from the very primitive living cells to the highly complicated living cells. In all of them, you will see that these biomolecules are important for various processes. So how many classes of these molecules are there? We have 4 major classes of biomolecules: proteins, nucleic acids, carbohydrates, and lipids. So look at the beautiful structures that they have. Now, starting from proteins, as the name indicates, proteose itself means that they are of primary importance. So proteins are very, very important because they carry out many important roles in the body. And proteins can take up very nice hierarchical structures. Then coming to carbohydrates, the moment we say carbohydrates, we remember energy because we remember glucose. So the major role of energy production lies in carbohydrates. And these are our wondrous molecules, nucleic acids. They are the genetic material. Now, coming to lipids, you should understand that from a unicellular organism, in the process of evolution, we have become highly complex multicellular beings, probably because of what we call it as compartmentalization. Compartmentalization is where you have separation of cells from one to multicellular, unicellular to multicellular. And that is probably because we have a very dynamic biomembranes, and these biomembranes are made up of lipids. So every molecule that you see here, all the 4 classes of biomolecules share that important responsibilities of making our cells alive, dynamic, and extremely functional all through the cell cycle or all through the life cycle of an organism. So we will try understanding each one of them in whatever little detail possible in this a few minutes of time that we have, because otherwise if you go to any science library, you will see that you will have volumes and volumes of books on each of these biomolecules, because understanding their intricacies and understanding their detailed structures is by itself a whole new subject. Okay, right. So starting about proteins, as I said, initially, if you look at how the biomolecules were understood, identified, evolved, proteins were the first ones to have been understood very clearly. So the entire initial journey of biochemistry as a branch of science started with proteins. Most of the scientists attributed many important functions only to proteins. So they are the fundamental building blocks of our body. And a very important point that all of us should remember is all these biomolecules have monomers. What is the meaning of a monomer? These are the individual units which collectively interact together to form biomolecules or to form the macromolecules. So all your biomolecules are basically macromolecules which are made up of monomeric units. So the monomeric units of proteins are called as amino acids, which by themselves have a very unique structural designing, which we will talk about a little later. Now, they perform almost all major functions in the body, if you remember: bodybuilding, growth, repair, hormones, enzymes, transport proteins, regulatory proteins. Almost all important functions in the body are carried out by proteins. And the very interesting thing about these proteins is they're made up of amino acids. Now, when the amino acids are joined by peptide bonds, they form macromolecular structures. And because of this alignment, they can take up a structural hierarchy. Now, this structural hierarchy of proteins ranges from the primary structure, which is the basic. Primary structure is just the linear sequence of amino acids, to the secondary structures where they can take up some local alignments like alpha helices or beta sheets. Then you have tertiary structures, which are the 3D images. So that is like in a tertiary structure, you will see that some amino acid, which is, let's say, far from one another, an amino acid at position 10 comes close to an amino acid at position 110. Like this, you will see when they fold into three-dimensional structures, they bring amino acids which are otherwise farther from each other close together. And most of your enzymes, globular enzymes, digestive enzymes, they all belong to this hierarchy. That is a three-dimensional structures. Why? When they form a three-dimensional structure, it is possible for the proteins to have active sites in which the substrates can come and bind. So based upon the reactivity, if that particular protein has to be designed into a tertiary structure or into an enzyme, it will take up a tertiary structure. Then the beauty of proteins is understood when you look at the quaternary structure, where you will see that multi-subunits interact with each other, forming a quaternary structure. Now, the classic example of the quaternary structure of a protein is hemoglobin, which has 2 alpha chains and 2 beta chains, which interact to form a cooperative molecule called as hemoglobin. And if you look at the process of evolution, you will see that in multicellular organisms with huge body, the dispersion of oxygen from very high partial pressure to very low partial pressure is possible because of this multi-subunit alignment of hemoglobin. So what is the basic understanding that we need to have? After the proteins are synthesized in the process of translation, they don't immediately become functional. Depending upon what role they have to play and what organelle they have to reach, there are post-translational modifications which decide the composition and the modifications of those amino acids. Based on that, the protein will get its unique composition, and based upon the sequence of amino acids, these proteins can take up these structural hierarchies. So proteins are functional either in their primary, secondary, tertiary, or quaternary structure. Now, in whichever structure the protein is able to perform its function, we call that as the native structure of a protein. Native structure is the structural hierarchy in which the protein is actually functional inside the body is called as a native structure of a protein. So that can either be primary, secondary, tertiary, or quaternary. Now, we are saying that these proteins can take up these nice 3-dimensional and 4-dimensional arrangements, all because of the presence of the monomeric units called as amino acids. And amino acids are the most unique molecules in nature because there is no other chemical substance which can hold one amino group and one carboxylic group in one structure. So amino acids basically have one alpha carbon atom, which you see is the central carbon atom, that has one R group, which decides the nature of the amino acid. And for every amino acid, there is one amino group and there is one carboxylic group. Now, because this central alpha carbon atom has 4 different groups on 4 positions, we call that as the chiral carbon atom. So, all amino acids except for glycine. Understand that glycine is the simplest amino acid, which also has its R group is H. So, that becomes CH2. So, except for glycine, all other amino acids have a chiral carbon atom with 4 different groups on its 4 positions. And these amino acids, because of the presence of the amino and carboxylic group, they can form a unique amide bond that is called as a peptide bond. So you will see the carboxylic group of one amino acid will form a linkage with the amino group of the second amino acid, resulting in the formation of what is called as a peptide bond. So when 2 amino acids combine with one peptide bond, we call it as a dipeptide. So like that, you have tripeptides, tetrapeptides, and when you have large number of amino acids joining together, we call them as polypeptides. And many such polypeptides together can form the quaternary structures of the proteins, or they can take up the sheet-like structures, helical structures. And in all this, the peptide bond is the one that is rigid, and the bonds that are present on either sides of the peptide bonds are flexible in certain angles, resulting in the formation of 2-dimensional, 3-dimensional, and quaternary structures in proteins. So ultimately, how many amino acids do we have? We have a total of 20 standard amino acids. When I say standard amino acids, these are the amino acids which come via the process of protein synthesis. Quickly, let me review. I hope all of you know what the central dogma of life is. What is a central dogma? Central dogma of life is DNA gives rise to RNA, and RNA gives rise to proteins. So DNA undergoes self-replication, and DNA carries out transcription to produce RNA. And from RNA to protein, we call the process as translation. Of course, we later identified reverse transcription, where RNA also can give rise to DNA. So this is a central dogma. So in the original genetic process where RNA undergoes translation to gives rise to proteins, there is what we call it as a specific genetic code, which codes for these standard amino acids. So all these 20 that you are able to see on the screen are called as standard amino acids. And what is different from one amino acid to the other amino acid is their R group. R group, remember, is the one that is connected to your alpha carbon atom. Now, this R group can result in the classification of these amino acids into different classes based upon what is their nature of that particular R group. If the R group is a neutral, then we call those amino acids as neutral amino acids. Now, most of the amino acids that you see here, glycine, alanine, valine, leucine, isoleucine, all these have long aliphatic side chains without any charge on them. So we call them as aliphatic neutral amino acids. They don't have any charge, and most of these amino acids are aliphatic because they have long carbon chains in their side chains. Whereas amino acids like serine and methionine, they have, methionine has sulfur in it. Similarly, cysteine has a sulfur in it. So we call cysteine and methionine, because they have sulfurs, we can call them as sulfur-containing amino acids. So like this, depending upon their R group, we can divide them. Now, amino acids, phenylalanine, tyrosine, and tryptophan, you look at their structures, you will see that all three of them have cyclic benzene rings in their structures, because of which they are called as aromatic amino acids. Now, proline is one very unique amino acid. You see that this is the complete structure of proline, where the main chain amino group is in linkage with the side chains. That is the reason why proline is called as an amino acid instead of an amino acid. So when you do classic tests for amino acids, proline is the only one which gives a different color, right? Now, all these amino acids that we are talking so far, they all have groups. Some have neutral, some have polar groups, but they don't carry any charge. But those amino acids which contribute to the charge on a protein, we call them as polar charged amino acids. In polar charged, you have two types. Those that contain an extra carboxylic group are called as acidic. Those that contain an extra amino group are called as basic. So, aspartic acid and glutamic acid are considered as acidic amino acids, and they do carry one extra charge carboxylic group. Lysine, arginine, and histidine are considered as basic amino acids. So, overall, the 20 amino acids, based upon their R group, they're classified into either neutral, polar uncharged, polar charged. And the classic example is aromatic amino acids, which carry benzene rings. And generally, when you look at the analysis of proteins, these aromatic amino acids have a maximum absorption at 280 nanometers. That is the reason why proteins, spectrophotometrically, if you're analyzing, you will see a peak at 280 nanometers corresponding to the presence of aromatic amino acids. And most of the proteins carry some amount of aromatic amino acids in their structures. So like this, there are 20 amino acids. All of them have a specific genetic code. Apart from this, you should understand that there are a variety of derived and non-standard amino acids, those that actually contribute to the variety of the proteins. Because if you look at how many proteins are available in nature, with just 20 standard amino acids, we already know in a protein data bank, we have about more than 1 lakh proteins that are identified. Above this, there might be so many other proteins in the organisms which are still yet to be discovered. So the question here that we have to ponder is how just these 20 amino acids are contributing to so much of variety. How with just 20 amino acids, you're able to produce thousands of varieties of proteins, and the amount of proteins that are produced in just one human body are in thousands. So how is it that we are able to produce? That's because of the post-translational modifications, which results in the production of what are called as non-standard or derived amino acids like hydroxyproline, hydroxylysine, methylarginine. So there are some modifications that are done that add to the variety of the proteins. So this is like an ocean of understanding. You start talking about the structure of proteins, you go into the details. It is so fascinating to know how these amino acids can design themselves so as to attribute a particular function to a protein. For example, you have proteins like collagen, which are known for their tenacity and strength. At the same time, you have proteins like elastin, which are very soft and tender. Same time, you have proteins like pepsin, chymotrypsin, which work in an extremely acidic environment in your stomach at a pH of about 1 or 2. Right? And so like this, there are enzymes which can work with, lakhs and lakhs of substrate molecules being converted per unit time. So we are creating a variety with just these 20 amino acids and their modifications. So we should say, hats off to all these proteins, all these amino acids, which are able to undergo that modifications to create so much of variety in life. So that's about very brief introduction and understanding about proteins. So what are the important lines that you should remember when you're talking about proteins? Proteins are very important. They carry out many major functions like being first enzymes, hormones for growth, repair, reproduction, transport proteins. They carry functions. And what are they made up of? They're made up of amino acids. What are amino acids? They have a basic alpha carbon atom, amino group, carboxylic group, R group. Based upon the R group, the amino acids can be divided into neutral, polar charged, polar uncharged, and they can attribute to the functions of the proteins. And also there are a variety of non-standard and derived amino acids that are important for the formation of protein structures. And later, when you are understanding, there is so much of in silico analysis in biology. There is a field of science called as bioinformatics and computational biology that is picking up, where you will have to do all these studies and analysis computationally also. So remember, every amino acid that is adding up to a protein has a particular, this is its full name. It has a 3-letter abbreviation and a single letter code. So all the single letter codes of the protein amino acids are important, because when you look at the sequences in databases, you will see them as G, A, L, 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 I, P, P. What are those alphabets? They correspond to the particular amino acids. So for most of the amino acids, their name, whatever is the first alphabet of its name, also is its single letter abbreviation, except for some where, suppose, let's say for proline, P is already used. So therefore, phenylalanine has an F. Like that, you have 5 or 6 deviations. Otherwise, most of the amino acids have their first letter as their single letter abbreviation. So an awareness about the single letter abbreviations also is important for your futuristic perspective of biology. Right. So we understood proteins. We understood what is a central dogma. So we said all this beauty of proteins, their intricacies, their structures is all possible because the proteins are synthesized from the master genetic molecules called as nucleic acids. So nucleic acids are the ones which actually carry the entire genetic information. Okay. Now, as I said, when scientists across the globe started understanding biochemistry as a branch, their first understanding is proteins are the molecules of primary importance. So almost proteins do everything. So initially it was understood that even the process of heredity and genetic information also is carried by proteins. But later in 1930s and 40s, scientists started doing a lot of research as to what are these molecules that are associated with carrying of genetic information or heredity. Then the experiments done by various scientists, starting from Griffith to Avery, MacLeod, McCarthy, they all have proven that it is indeed nucleic acids that are able to act as genetic material. So nucleic acids, the name, because they have a phosphoric acid molecule in their structures. So these are the only biomolecules which have an acid content, and they were identified for the first time from the nucleus of pus cells. So therefore, they carry the name nucleic acids, that is acidic molecules that are present in the nucleus. So we know there are 2 important types of nucleic acids, DNA and RNA. You should remember that in the entire process of evolution, now of course it is all a hypothesis because we cannot go back to millions of years ago and identify what it is, but the entire initial process of evolution, RNA is the genetic material. So we know that most of the single-stranded viruses have RNA as their genetic material. From single-stranded viruses, when the process of evolution got into higher order organisms, RNA is not very stable. Therefore, the responsibility of acting as genetic material is transferred to DNA. So from the double-stranded viruses, you will see DNA is acting as a genetic material. Only single-stranded viruses have RNA. From them, DNA started acting as a genetic material because of the lesser stability of RNA. Now we will understand why RNA is less stable. So now the whole job of carrying genetic information, heredity is all under the aegis of DNA. So what does RNA do? RNA is still a very responsible sister molecule of DNA, and the entire process of transcription, translation, that is the molecular expression, is all vested upon the RNA molecules. So DNA does this job of carrying genetic information in the nucleus. It acts as a hereditary material. Whereas RNA does all the molecular processes, that is transcription, translation. So you have the 3 types of RNA molecules, mRNA, rRNA, tRNA, which are very, very vital for the molecular expression systems. So whether it is DNA or RNA, what are they actually made up of? So all nucleic acids have 3 important components. One is a sugar molecule. Plus they have what are called as nitrogenous bases. Then they have a phosphate molecule. So this we call them as nucleotides. Nucleotides are the monomers of nucleic acid. So what are the monomers of proteins? Amino acids. What are the monomers for nucleic acids? Nucleotides. What are these nucleotides made up of? Sugar plus nitrogenous bases plus phosphate. So nucleotides are sugar plus base plus phosphate. Now we have many active monomeric units of nucleic acids that are also functional in the body, which we call them as nucleosides. They don't have a phosphate, they have sugar and base. Now our favorite nucleotide that all of us know is ATP, adenosine triphosphate. This is the most important energy rich molecule in the body, vital for all metabolic pathways. So understand that apart from just acting as genetic material, nucleic acids also carry this additional work as nucleotides and nucleosides. They carry many major responsibilities inside the cells, most importantly being energy carriers. Now, nitrogen bases are of two types, purines and pyrimidines. So, adenine and guanine are purines. If you look at their structures, you will see they have two ring structures. So they have one cyclic benzene ring and one imidazole ring. So adenine and guanine are purines. Thymine and cytosine are pyrimidines. So we have two purines and two pyrimidines in DNA. Whereas in RNA, in place of thymine, the pyrimidine you will see is uracil. So adenine, guanine are common both in DNA and RNA, whereas thymine is present in DNA, whereas uracil is present in RNA. And when you look at the sugar structures, you will understand the DNA is 2-deoxyribose, that is in the second position of the ribose, there is a deoxy, whereas RNA is a ribose sugar. Now, when you look at the sugar structure, because of the presence or absence of a 2' hydroxyl group, this is your 2' carbon atom, RNA has this 2' hydroxyl group, making it more susceptible to chemical reactions. That is why we said RNA is less stable compared to DNA. So RNA's sugar has a 2' hydroxyl group, which is chemically very reactive. So what happens when you start making RNA strands? The 2' hydroxyl group starts internally cyclizing, resulting in lesser stability of RNA. So DNA is more stable as a polypeptide structure compared to RNA. Therefore, RNA carries out all other additional responsibilities as mRNA, rRNA, and tRNA. So what is the outline understanding of nucleic acids? Very important as carriers of genetic information. In viruses, we have RNA. Rest all organisms have DNA as a genetic material. RNA is less stable because of the 2' hydroxyl group in its sugar. Now, they are made up of sugar, base, and phosphate. And the linkage between these two nucleic acids is called as phosphodiester linkage. Now, we move on to the next important class of biomolecules called as carbohydrates. Very important, our favorite molecule, that is glucose, is a class of carbohydrates. So what do these do? Basically, they are associated with energy production. Almost all metabolic networking is because of the presence of carbohydrate molecules. And these carbohydrates basically can be divided depending upon the number of monomeric units. So the monomeric units of carbohydrates are monosaccharides. So these monosaccharides cannot further be broken down into any further units. So they are called as monosaccharides. So depending upon the functional group, the monosaccharides can either be aldoses if they contain an aldehyde group, or ketoses if they contain a ketone group. So we call basically carbohydrates, chemical definition, polyhydroxy aldehydes are ketones. We call them as polyhydroxy aldehydes are ketones because they carry aldoses or ketone functional groups, and they're very important for the process of energy production. So if they have two such monomeric units, we call them as disaccharides. If they have many such units, we call them as polysaccharides. And our most important polysaccharide we know is starch. So starch is a very important polysaccharide in plants. And the most important polysaccharide in animal cells is glycogen. So in animal cells, you have glycogen. Whereas cellulose is very important, which is a important polysaccharide you will see again in all plant cells, that which cannot be digested by animal cells. But starch is the most abundant polysaccharide because this is the one that is synthesized from the process of photosynthesis. Starch is abundantly available carbohydrate. Apart from just providing energy, apart from just being the molecules providing energy, ATP synthesis, citric acid cycle, glycolysis, all metabolic pathways associated with carbohydrates, they also carry other important functions. Like we have a very important class of carbohydrates called as glycoproteins, where they combine with proteins. We have proteoglycans, where they combine with glycans, that is polysaccharides, resulting in the formation of complex structures, blood group substances, membrane structures. So carbohydrates play various other important roles. Last class that we're talking about, lipids. Now, all these classes that we have discussed so far, that is proteins, nucleic acids, carbohydrates, all the three are water-soluble biomolecules. It is only lipids which are a very heterogeneous class and water insoluble. That is, lipids are a completely organic molecules. They are non-polar. That means they are not soluble in water. That is because they have very long chain fatty acids as their monomeric unit. So what are the monomers of lipids? Fatty acids, and they are non-polar, water insoluble. Now, depending upon their structure, lipids again are a very complex class of molecules, but it is their non-polarity that is very important for the formation of biological membranes. You remember in the starting of the presentation, I told you how compartmentalization and the evolution from unicellular to multicellular is possible because of the membranes. So they help in compartmentalization, and they result in the semi-permeability of the biological membranes exclusively because lipids are non-polar. And surprisingly, the lipids can be classified as two. We can call them either as structural lipids or storage lipids. So the moment we say lipids, we use the word fats. Triglycerides can also be called as fats. So this is one word that really scares all of us because fats result in obesity. But otherwise, apart from just adding up to your adipose tissue, fats have many important roles to play inside the body. So fats or triglycerides are an important class of storage lipids, same as with waxes, and they contain glycerol and fatty acids. So you will see that fatty acids are the monomeric units in all classes of lipids. These fatty acids, depending upon the number of bonds they have, saturated, they can be divided as saturated, unsaturated. Saturated means they don't have any double bonds, no double bonds, whereas unsaturated have double bonds. Depending upon the number of double bonds, if they have only one double bond, we call them as monounsaturated. If they have many double bonds, we call them as polyunsaturated. Now, all of you can do this. Go back, look at the oil packets that you consume in your day-to-day life. You will see that the oil packets are written enriched with PUFA. PUFA means polyunsaturated fatty acids, which are good for your health. So the more saturated fatty acids you take, the more adipose tissue deposition. But if you take oils which are rich in polyunsaturated fatty acids, they're good for your health. Then you have compound lipids like phospholipids, glycolipids, sphingolipids. They all are very important structural lipids because they are the ones that are associated with your membrane formation. So we either call them as membrane lipids or structural lipids. They are very important, complicated class of lipids, but they are very important. Your entire myelin sheath of brain contains these phospholipids, cerebrocytes, and gangliocytes. And you will also see that there are so many diseases associated if these lipids are not properly synthesized and assimilated inside the body. Then the last important class of lipids are derived lipids. Very important, we know steroids, And sterols. Again, this word cholesterol is an alarming one. We know high cholesterol is dangerous in the body, but minimal amounts of these lipids also are very important for the smooth functioning of the cells, because most of your steroids, like your sex hormones, bile acids, they're very, very important for the metabolic processes. So even some amount of derived lipids are required by us through our diet. So ultimately, biochemically speaking, all these biomolecules, because of their structural intricacies, are very, very important for the functionalities of the cells, starting from their basic metabolism to their higher level complex expression systems. All biomolecules work in unison. And see, this is where you can see these lipids have very long chain structures. That is why they are ultimately non-polar molecules. But when you look at the phospholipids, you will see that they have a hydrophobic tail, but they also have hydrophilic heads. Because of this amphipathic, so we call these molecules as amphipathic or amphiphilic molecules. That is, they're both hydrophobic and hydrophilic. So when they form a lipid bilayer, or hydrophilic heads are present on the outer surface, all hydrophobic tails are pushed into the inner side of the cells. This is called as a micellar structure that is formed with lipids. I will give you a small example for understanding. Now, when you use detergent, when you add a detergent or soap into water and you mix it, you get those small bubble-like structures, right? So those bubbles are nothing but micellar structures. So when you use a soap to clean dirt or clean your body, why do you think the dirt moves out? Because this soap or detergent, which is also a fatty acid molecule, has ability to form micelles. And when they form micelles, they carry all the dirt inside the micellar structure. And when you wash them, they go away along with the micelles. Now, this ability of lipids to form micellar structures is very important, because they form what are called as lipoproteins. Very important when if you ever get done, or if you see some of your family members are getting any diagnostic tests done, you will see that lipid profile is a very important diagnostic test that is done. It is a tool to identify any kind of disease conditions. So we have lipoproteins like LDL, VLDL, HDL. All these lipoproteins are formed because of the ability of lipids to form micellar structures. They carry along with them cholesterol, fat, fatty acids, all of them. What happens in our, why are we scared of taking excess fatty food? We all know, right? Whether we know the biochemistry or no, we know the nutritional importance of fats. What happens when you take excess of them? If you don't need so much of energy coming from fats at a particular point of time, they go and get stored as adipose tissue. So if the body doesn't have enough exercise or enough metabolism to break these lipids or these fats, then automatically it leads to obesity. So fats are a very important class of lipids, and mostly colloquially when we say lipids, we understand they're fats. But apart from the storage fats, there are so many membrane lipids also, like phospholipids, which are important. So to conclude, this is a very important, interesting slide where on the same thing you can see all the four comparison. So you can see this is important for you to understand what are the monomers of each of those biomolecules, some examples, what are they made up of, and what are their important functions. So if you can assimilate this information from this video, I think then that's the success of the whole story, because ultimately a clear understanding of what biomolecules are made up of and what do they in the body, what do they do in the body is very important for us to know. So carbohydrates, basically the monomeric units are monosaccharides, and what is the characteristic linkage? That's a glycosidic bond. Then you in proteins, the monomers are amino acids, and their characteristic bonding is called as a peptide bond. So in nucleic acids, I said their characteristic linkage is a phosphodiester bonds. In fatty acids, the bonds are ester bonds, but these are more heterogeneous. There are varieties of other bonds also in lipids. So these phosphodiester linkages in nucleic acids, peptide bonds in proteins are those characteristic linkages which stabilize the biomolecules in their higher order structures like secondary or tertiary structures. So carbohydrates, the classic examples we know, starch, cellulose, glycogen, these are all examples. As I said, collagen, insulin, keratin, very important. We know protein that is associated with hair, nails, that is keratin. Of course, DNA and RNA for nucleic acids, fats, oils, and waxes in lipids. And you will see, as I said, they're all made up of the basic elements, carbon, hydrogen, oxygen. In fact, there even sulfur and phosphorus are present in some higher order carbohydrates, like, as I said, glycoproteins, sulfated glycoproteins, they are present. All these elements are present in proteins, carbon, hydrogen, oxygen, nitrogen, phosphorus in nucleic acids, carbon, hydrogen, oxygen in lipids. So carbohydrates, first we said, is energy sources. Apart from that, they also have structural importance. Remember, I said glycoproteins, proteoglycans. Then, of course, they are the reserve food. So what happens? See, we are not constantly eating all the time. So when you consume something, then whatever is required immediately for the production of energy is used as glycogen, is broken down into glucose by various pathways like glycolysis and TCA and electron phosphor transport chain. But if there is excess of glycogen, then it is transported and stored as glycogen reserves inside the body. So not just instant energy, but as storage forms of energy also, carbohydrates are important. Proteins, of course, name any important function, protein. So proteins are my favorite molecules. They do a lot of important functions. Genetic information, nucleic acids. We also said other important roles like energy, ATP production. They help in the, as energy currency. ATP is called as the energy currency of the cells. Then lipids, first very important thing, are membrane components. Apart from that, they also act as energy source, because you all must have studied from the nutritional perspective that lipids have a sparing action on carbohydrates. So if carbohydrates are not available for supplementation of energy, it is immediately the lipids that we use. So what happens when, or let's say you're all dieting or you're on a diet, what is the first thing that happens? Your glycogen reserves are easily destroyed because they are with lot of water. Whereas lipids are tough. Even with lot of exercise, you only start toning, but your weight loss starts much later. Why? Because when fats are stored, they are a very concentrated sources because they are water insoluble. So when a fat is stored inside the adipose tissue, it is completely fat. There is no water. But when glycogen is stored, it is stored along with a lot of water molecules. So nutritionally, your diet is very important for us to produce all these biomolecules, because through diet, we consume proteins, we consume carbohydrates, we consume fats. So all these also have a lot of nutritional importance. So these fats are concentrated sources of energy. So when carbohydrate is not available, a classic example that you can take, suppose, let's say, one person is under starvation. Now what happens is his glycogen reserves can support him only for 1 day or 2 days or maximum 3 days. But after that, if still the body is not getting any glucose from the diet, then automatically the cellular metabolism relies on lipids for energy production. Now, in worst case, if the person is under continuous starvation, you become completely lean, all your fat resources are used. The last is muscle wasting, where even proteins can sometimes be used as supplementation of energy. And you will see there is a complex metabolic networking inside the biological cells, and you will see that all these biomolecules work in unison. So you take a unicell, single cell, is like a guru. It will explain you all the important rules of coexistence. So that starts first with your biomolecules. So none of these biomolecules work independently. They are always together. So for a molecular expression, if DNA has to work, DNA-protein interactions are important. Until and unless DNA dictates, proteins are not synthesized. Until and unless the proteins are expressed and they carry out the functions, you don't know how much energy you need. So therefore, they rely on carbohydrates. Now, to do all these things, membrane dynamics are important, which are given by lipids. So you see that all biomolecules are extremely coordinated. There is a wonderful chemical coordination and coexistence, which is helping us to survive without major ailments and a proper metabolism, even when we are sleeping, when we are alive, when we are awake, when we are eating, when we are not eating. Nothing in your body is hindered. All the metabolic processes go on so smoothly because of the coexistence or co-working of these biomolecules, which tell us a very important lesson that we should learn being such small molecules, being made with such small basic elements, they are creating so much of variety, they're creating so much of functionality. So let us all learn that biomolecules teach us some very important lessons of life, apart from just maintaining the symphony of life. So any doubts and questions about biomolecules, you can post here, or you can further write in the chat box. We can discuss on that. So thank you all. So that's the end of our discussion on biomolecules.